Patrick Liu. He's coming from the beautiful Guangzhou, from near the Li River, one of the most beautiful places of China, and therefore he missed his morning presentation, and he will give us a presentation about peptide-based nanoparticle for targeted delivery of siRNA.
Thank you. Okay, sounds like it's working. First of all, I would like to say sorry about I missed the morning session, but I hope for the following presentation I can compensate that shortcoming. However, looks like we already heard something more traditional drugs with new delivery systems. What I'm going to present to you is using new modality of drugs like siRNA with a novel nanoparticle delivery system to address unmet clinical needs. So more about what's the dynamic of the RNAi therapeutic field rather than the detail of the data I will going to present to you due to the limitation of the time. Okay. So here, just give you a quick overview of what's going on. RNAi therapeutics, we all know 10 years ago it's become very hot. Science magazine gave RNAi like a breakthrough of the year, and in 2006 we have Nobel Prize. Big pharma all jump in. And then after 2008, things a little bit cooling down. And for last 5 years, people say, hey, RNAi may not be that promising. However, since 2 years ago, the things turn around. As what you can see, this particular article published in last month's Nature indicated that RNAi interference rebooted based on many successful clinical studies. Currently, although we don't have approved products yet, but we have many clinical trials ongoing. Just look at the financial market. It's also very active right now. On Island, the leading siRNA therapeutic company, valuation currently about $5 billion without any marketed drug. And another example is siRNA. They also just have one of the phase 1 trial. They went public in February. Their market valuation at the time is $600 million US. So just give you some idea what's really going on in both scientific field, clinical field, and financial market. And here, pretty much the overview about current clinical studies ongoing. You can see through oncology, infectious disease, and some other unmet clinical needs. For example, wound healing, and also scars, and influenza, and other respiratory diseases. So I have almost 20 years industry experience in developing nanoparticles for therapeutic development, use nucleic acid as drug modality. First 10 years working with Novartis for gene therapy effort, and since 2001 we started working on siRNA therapeutics. The first company I co-founded with another colleague of mine sold to Genentech lead venture groups 2006, and then I started a second one called Sonomics, and also we have subsidiary in China 6 years ago. So our strengths, it's more based on the delivery, use polymer-based nanoparticle. As what you can see, siRNA therapeutics, because the mechanism of action is in the cytoplasma, so you need delivery, the drug material into the cells. On the other hand, it's definitely a little bit easier than gene therapy because you don't need to get your material into the nucleus, and you don't have those kind of potential toxicity or long-term damage of integration of those nucleic acid into the genome. So RNAi clearly has its advantage. However, it's a need delivery. The delivery can be enhance the endocytosis and also need to be released from the endosomes and then allowed RISC complex to really work with the drug material to break down the target sequences and have the therapeutic benefit. Currently, siRNA regulated by CDER, not CBER, by US FDA. Similarly, in China, also regulated as a small molecule drug. So that's somewhat different from gene therapy and other biologics. Even though we use the nanoparticle polymer, sometimes we put the ligand on top of it. It's more complicated than the simple small molecule drugs, but they still put us as small molecule entities, but with support from CBER-like function. So the current most successful siRNA therapeutics coming from Alnylam and Takmir, basically they all use lipid nanoparticle. This lipid nanoparticle actually is quite complicated than the traditional simple lipid nanoparticle composition. This lipid include PEG and also have cholesterol and other components, basically 4 or 5 components in this lipid nanoparticle. They already developed very good process for this, and Alnylam already have their phase 3. That's why their market valuation is so high. And Takmir just finished 2 of their phase 1 study. So why their valuation is so high? One way to see it, because they have a delivery system. Some other companies, in comparison, ISIS, have long-term development, has more than 16 phase 1, phase 2 trials ongoing, but the valuation is still below, very much below Alnylam. Why? Because the anti-science first, second, they don't have very strong delivery vehicle. So delivery is the key, again. So we work on delivery for quite years, as what I already talked about. Not only in US for last 15 years, we also push our effort in China. So enhance siRNA therapy development and develop our peptide-based siRNA therapeutics. So as a biotech company, as what you all know, IP is very important. On the other hand, for us, we are not only have IP, PCT protect from the global point of view, we also need to develop some local Chinese-started intellectual property, mostly because we want to help the local government to have their innovation kind of thinking and mentality. So Sonomics and Nanotides, it's the same company but with different functioning groups because of different geographical location. We have 3 generations of nanoparticles for siRNA therapeutic development. The first generation, basically, it's our common used systems, positive charged polymers or positive charged liposomes in combination with siRNA. It can self-assembled into nanoparticle, and it can be easily formulated, as what we just heard the previous speaker. The very efficient development of those nanoparticles going to have a much better application, especially for the developing country patients. And we currently, for our current indication, we use this particular one, HKP. That's a polymer-based. H stands for histidine, K stands for lysine. It's a lysine histidine copolymer, originated from Professor Jim Mixon in University of Maryland. That's more than 20 years ago. We start collaboration with Jim. when I was still with Novartis 20 years ago, when he was with NIH, and we keep that kind of a relationship for the last 20 years. We still work with him. So right now the technology already moved to second generation and third generation. I will give you some overview on that. So this is the second generation. What the second generation is, basically adding active ligands. That ligand can enhance the active targeting. I think most people very well know about it. So what we did, we have a specific peptide isolated from malaria protozoan parasite. So take that peptide, covalently bonding to HK polymer, use that formed nanoparticle. We can see here have very good hepatocyte-specific targeting. And this is a HepG2 cell as well, compared to this one is A549, it's a lung cancer cell, and this two doesn't have that ligand. So this is one example. You can enhance the liver targeting. We also use a PCAM protein antibody. This is in collaboration with Professor Sung Lee from University of Pittsburgh. He used this antibody in combination with a lipid nanoparticle, can systemically deliver into the mice model, but achieve the lung endothelial layer targeting. So it's systemically delivered, reach to the lung. And this is our early work, 2004, we published this work. I think we were the first group published a nanoparticle systemically delivered with RGD as ligand, reach to the tumor neovascular surroundings. That result tumor inhibition through minimize or downregulate angiogenesis around the tumor stroma region. And currently we further improve. Rather than use PEI at the center condenser, but we use HK polymer. That work just published last year. So about 4 years ago, we worked with another group, started talking about use the upconversion material. I think we do have other presenters talk about upconversion material. Basically, NCI asked if anyone can develop a nanoparticle, can enhance the imaging, same time can deliver drug to accelerate therapeutic effect. So that's the system maybe have that kind of advantage. Basically, the near infrared can activate this upconversion material. We synthesize in our own labs, and we can activating those surfaces of this with this silicon shell and incorporate with certain kind of photosensitive drug. And then we activate the surface with the amine groups. We can have siRNA and the peptides. The peptides can be more targeting effect, and the siRNA can enhance those small molecule drug and singulate oxygen effect, anti-tumor effect. So that's the idea. And then as what you can see for in vitro transfection, without any other delivery vehicle, the labeled siRNA almost 90 to 95% transfect into those two different cell types, 293 and HeLa, I think. And we also test in vivo. So this is the third system. However, this system is much more complicated when we do the development. Currently, we are using the first generation system for our clinical viable study. So this is our clinical program. As what I already indicated, delivery is very challenging. So rather than go through oncology, although we have quite many NCI grants, but we go through the surfaces, local surfaces. So we go after scar. Scar issue is still a very challenging issue for clinical. There's no FDA-approved pharmaceuticals currently for scar treatment indication. So it's a huge market. We all know about it, but we don't have good therapeutics and good pharmaceutical address that issue. So we work with use siRNA, target TGF-beta1 and COX-2. I will give you some details. And to develop siRNA therapeutics, use peptide copolymer-based nanoparticle. We also use siRNA to address influenza. Influenza, it's going to be constant problem now and in future. So we are currently working on that program too. So you can see we have a long list. I will just give you some quick example about our first two programs. So this HK polymer we are using for topical delivery address this scar problem. So here is typical siRNA HK polymer nanoparticle. We lyophilize it. We also formulate with the microcellulose and even aqueous solution. We have used this for tumor systemic delivery and also for lung delivery, that's for influenza. And this is for skin topical wound delivery. So this is the first products we are currently filing IND in China. This is the #1 siRNA therapeutic program in China, and the first filed. We also file in US at the same time. We are filing IND in US right now. So as what you can see, the market potential is tremendous. And there's so many surgeries, all different kind of surgeries, and even traumatic wounds. For example, car accident and other burns, a lot of requirement. So we target TGF-beta1 and the COX-2, and use our structure. It's 25 more siRNA rather than 19, 21 that specific more, and siRNA 27 more, and some other antisense related structures. We use 25 more blunt-ended. So we believe this is clearly has a very smooth IP maneuver to other blockers. And we already complete all the IND-enabling study. And we have partnership in China, and also we have a clinical collaboration in both China and US with Johns Hopkins. So here I can show you some of the data. So at early stage, we work with mouse model. And this, the question we ask first, although we want to improve the scarless condition, if that drug will slow down the healing process. Here, just want to demonstrate with this kind of treatment, the healing process not only is not slowed down, but still slightly enhanced. Here, just want to show you the histology evidence. Use the combination of TGF-beta1, COX-2 knockdown. At the same time, you achieve the structure very much like a normal skin structure. We have more data to show that. And with a larger wound site, it's also improved the wound closure. And with diabetic mouse, it's also improved wound closure. And then later, all those effects relate to the downregulation of two genes, TGF-beta1 and COX-2, have dose-dependent effect and also have time differences. And then we move to a peak study. And then this is a peak accessional wounds study. This is a peak burn wound study. You can see this is a dramatic effect, both on the surface of the to observe the scar and also histology support. And last year, we have a group in China. They use a human scar. and implant into mouse. And we directly inject our STB-705 into those implant scar, the scar minimized. So that means the scar can shrinking. And we did a lot of study, ask what's going on there, why the scar can shrinking. So based on a lot of studies, we are currently putting a manuscript out. TGF-beta1, COX-2 simultaneously silenced have upregulated or induced apoptosis of fibroblasts. So fibroblasts is it's the cause of the extracellular matrix accumulation. It's cause of the scar. When you turn off or minimize the fibroblast proliferation and even apoptosis, you clearly can minimize scar formation. So that's the pretty much the data set we put together to support these products. And we complete this study. Ironically, in China, they even ask you to do much more than what US FDA is going to ask for siRNA therapeutics, for this kind of anti-scar therapeutics. As what you can see here, we have to do a 40 monkey study. So it's topical. And then this is indication going to be scar revision. The clinical study going to be both in Shanghai and also in Johns Hopkins. And one thing I want to point out, there are two competing candidates, drug also ongoing. One is Excalar, partnered with Pfizer. They use antisense targeting CTGF. And then what level they're using is 5 milligram per centimeter, per square centimeter, or per centimeter long if it's an incisional wound. RSI are also in the similar range. And what we use, it's almost 2 or 3 logs below their dosage. Why? Because we have nanoparticle enhanced delivery, much higher therapeutic efficacy. And we already completed all this study. That's also demonstrate the process development. And we have a large production in collaboration with a Chinese company, pharmaceutical company. And we have other products also moving ahead. This is influenza. I'm not going to spend too much time on that. And we have an oncology drug candidate also ongoing. So that's pretty much our big picture. And I would like to take this opportunity to thank the collaborators and people working in the lab. And also thank you for your attention.
Thank you very much, Alessandro.
Question? Yeah.
Thanks for this very interesting presentation. You mentioned that in China, these compounds are considered small molecules. So in the future, we will be talking about generics and not biosimilars. How does it work? I mean, you have...
What's the generic?
In the future,
okay.
In 20 years from now, another company will try to make a generic of your product. Mm-hmm. This will be considered a generic or a biosimilar?
It's not a biosimilar. It's not still generic.
It's a generic.
It's a small molecule. It's I don't think Chinese regulatory body try to do anything new. They just follow what US do. So all siRNA therapeutics in US is regulated by CDER.
Okay, thanks.
So I have a question to you. In the preclinical studies, when you treated monkeys, first, did you make a scar for the monkeys when you treated, or just put something on the healthy skin?
For preclinical study, we have to do multiple things. I didn't present for making scars. We have to make scars on pig and a guinea pig. So want to show the drug distribution, and also want to show the reaction, the skin reaction. So that's for specific skin-related therapeutic test. Mm-hmm. For monkey, it's basically a safety study. It's sub-Q injection, 3 different doses, and multiple individuals. So, and then 28 days.
So,
daily injection.
but you are planning to put this formulation on the scar, right? Just as a topical, not inject during the clinical
So
trial.
actually, we do have the data to support it either way. So right now, we made this dry powder, basically. It's injectable. So really depends if, so when we pass the phase 1 or the safety study, we will do at least 2 phase 2. One is injected, and then another one is topical applied. So
what is the major toxicity? What is what you see as major toxicity? Dose limiting.
Actually, there's no major toxicity, but potentially, if you repeatedly apply this peptide condensed siRNA, I worry about immunogenicity. That's from immunological point of view. Put on topical and then repeat putting there, and then doesn't matter. Even polysaccharides can induce some kind of reaction. So that's maybe potential problem. However, this wound, these scars will heal.
Yes.
So after heal, then you don't want to do anything more than that.
Much.
Yeah.
Thank you. Any other questions? No? The last speaker.
Thank you again.
Thank you very much. The last speaker is